Welcome to the Explainer. So imagine everything you learned in biology class about how life works. You know, the bedrock rules taught in literally every single science classroom for the last 70 years, just suddenly being flipped on its head. It's wild. Today, we're looking at a fascinating discovery from April 2026 that has biologists all over the world completely rethinking how genetic information flows. We're going to explore a microscopic defense system that breaks a rule we honestly all thought was unbreakable.
Okay, let's dive into this. To really grasp why this is so revolutionary, I want you to think about a giant factory. Can a factory machine, like the final product stamping out car parts on the assembly line, suddenly just pick up a pen and draw its own original master blueprint? Common sense, and honestly decades of biological science, say absolutely not. The blueprint dictates the machine, never the other way around. But what if a machine actually could do that?
Here is our roadmap for today's Explainer. First, we'll cover the golden rule, then move into the rule-breaking discovery. After that, we'll meet the Drt3 system, look at how proteins build DNA, discuss the viral defense decoy, and finally wrap up with the future of biotech. Let's kick things off with Section 1, the golden rule. You've probably heard of this golden rule. It's known as the central dogma of molecular biology, and it was proposed by Francis Crick way back in 1958. It basically states that genetic information flows in one very specific direction. Going back to our factory analogy for a second, step 1 is DNA. That's the master blueprint, securely locked away in the manager's office. Step 2 is RNA, which is kind of like the cheap, temporary working copy of that blueprint that gets sent down to the factory floor. And step 3 is the protein, the final machine built from those instructions that actually does the heavy lifting in the cell. The rule has always been super clear on this. Information flows from DNA to RNA to protein, period. Proteins absolutely do not pass sequence information back to nucleic acids, or so we thought.
Which brings us to Section 2, the rule-breaking discovery.
Get this, in April 2026, a team of researchers at Stanford University published a paper in Science magazine that literally pulled the rug out from under the central dogma. They discovered a bacterial system that actually uses a protein as a direct template to build DNA. Think about that for a second. The working machine was out there drawing the blueprint. Now, to be totally fair, biology always has a few tricks up its sleeve, right? We already knew about some minor exceptions to the central dogma. For instance, it's pretty well known that RNA can be copied back into DNA. Viruses like HIV use an enzyme called reverse transcriptase, which is essentially a biological translator working in reverse to do exactly that. But here's the catch. That is still nucleic acid to nucleic acid. The Stanford discovery is fundamentally different. A protein directly templating DNA synthesis? No way. That wasn't just considered rare, it was thought to be entirely, completely impossible until this paper dropped. Let's move right along to Section 3, meet the Drt3 system. To really understand Drt3, we have to zoom way in to a microscopic war zone. See, bacteria are constantly under attack by phages, and phages are simply viruses that specifically target, infect, and destroy bacteria. It's this endless, brutal evolutionary arms race, meaning bacteria have to develop some incredibly creative, extreme weapons just to survive these viral invasions. And that's exactly what the Stanford team, led by Alex Gao, was looking at. They were studying E. coli bacteria and uncovered this highly unique defense mechanism designed to fight off those invading phages. They named this defense system Drt3. It's basically a complex made up of 3 parts. 2 special enzymes, Drt3a and Drt3b, and a non-coding RNA molecule. You can think of that non-coding RNA as sort of a factory memo. It helps out, but it doesn't actually build a machine itself. Together, this whole crew teams up to build long strands of repetitive, double-stranded DNA. But honestly, it's how they build it that is just so remarkable. Section 4, how proteins build DNA. Now, what's really interesting here is how we shift from the what to the how. So the Drt3 system works in 2 very distinct steps. Step 1 actually follows the rules perfectly. The first enzyme, Drt3a, works the normal way. It looks at that non-coding RNA molecule, which acts as a standard template, and builds a single strand of DNA based on it. Okay, nothing crazy there. But step 2 is where things get absolutely wild. The second enzyme, Drt3b, has to build the matching, complementary side of that DNA strand, but it does this without any nucleic acid template at all. 0. No DNA, no RNA to copy from. Instead, Drt3b uses itself. Think of it kind of like a 3D printer. When a 3D printer creates an object, the physical shape of the printer head and exactly how it moves determines the physical output, right? Well, inside the active site of the Drt3b enzyme, specific amino acids act exactly like a physical mold. These amino acids physically enforce an exact alternating pattern, basically grabbing and guiding the DNA building blocks, the nucleotides, into their perfect, precise places. The lead researcher, Alex Gao, actually summarized it perfectly with this quote. He said, "The protein itself serves as the blueprint for the DNA sequence. It's just a fundamentally new way that life can produce DNA, and the researchers actually visualize this happening at near atomic resolution. It's brilliant." Okay, on to Section 5, the viral defense decoy. Now, you might be wondering, building DNA out of a protein mold takes a ton of energy and effort. Why even go through all that trouble? Well, it turns out the repetitive DNA strands that Drt3 pumps out act as a molecular decoy, or basically a giant sponge. When a phage attacks and tries to replicate, this massive web of repetitive, custom-built DNA interferes with the virus. It literally clogs up the phage's replication machinery, or it alerts other bacterial defenses. It's an incredibly successful survival strategy that has been evolving over millions of years. So the crucial point is this. We definitely need to make an important clarification on the central dogma here. This discovery doesn't mean that proteins are suddenly writing arbitrary genetic code everywhere all at once, or that cells are reverse translating their entire protein library back into DNA. The golden rule isn't destroyed, it's just expanded. Drt3 is a highly specialized system producing one specific repetitive sequence for defense. But conceptually, yeah, it is a massive paradigm shift. Finally, Section 6, the future of biotech. So why does understanding some weird bacterial defense system actually matter to us? Well, it matters for three massive reasons. First up, microbial surprises. It just reveals how much we still do not know about the microscopic world. Bacteria are full of biochemical surprises that completely defy our textbooks. Second is biotech potential, which we'll look at closer in a second. And third, evolutionary insights. It proves that the enzymes inside us are incredibly versatile and have been creatively repurposed by evolution time and time again. Let's zero in on that biotech potential for a minute. If scientists can figure out how to engineer that Drt3b protein, like if we can actually swap out the 3D printer head to make sequences other than just a decoy sponge, we could unlock a grand new tool for creating custom DNA. We're talking about advanced materials, programmable DNA hydrogels for medicine, and huge, revolutionary synthetic biology applications. Remember the world-changing gene editing tool CRISPR? Yeah, that also started out as just a simple bacterial immune system. Drt3 could very well be the next frontier. This discovery is such a beautiful reminder that biology is literally never as simple as our models make it seem. The central dogma is a highly useful rule of thumb, but life, especially in the relentless arms race between bacteria and viruses, will always find creative workarounds. So I'll leave you with this question to ponder. Knowing that biological machines can sometimes draw their own blueprints, does this change how you view the flow of genetic information? Biology is wildly inventive, and it's super clear we are only just beginning to scratch the surface. Thanks so much for joining me for this explainer. Keep learning, and I'll see you in the next one.